All right, we're finally going to finish up Module 10 here, Kingdom Fungi. Remember, we talked about the Basidiomycota last time, and those were the mushrooms, or the club fungi. We looked at some mushrooms, we dissected those, right? And so you should have something like this written down on that chart on page 80, yes? Okay, so today what we're going to do is we're going to go through the rest of these groups right here. Myxomycota, not so much, because remember we reclassified those as protists. Those are the slime molds. I will talk about them some more today because they're kind of cool. I think I even have a little video on them. So we're going to go through these, and as I go through them, you're going to write down a few things underneath there, okay? So let's go with Ascomycota right now. All right, example are yeasts and morels, single and multicellular. They're also known as sac fungi, SAC fungi, sac fungi. That's because they produce their spores in a little sac called the ascospore. That's the spore. The ascospore is made in the little asc right there. They're saprophytic. Remember what saprophytic means?
They eat dead things?
Eat dead things, good. Some are parasitic. You definitely remember what that means, right? They parasitize their hosts. They reproduce sexually by producing ascospores and asexually by budding. The daughter cell actually remains attached to the parent cell, unlike bacteria. We were supposed to try to grow a little yeast culture today, and I've had a pretty crazy day. My first class I had to start late because I forgot some stuff. I had to run back home and get it. Second class I completely forgot the lab. Last class we didn't get the math finished on this, but that's okay. And I don't have any yeast, but I do have other molds for us to look at, so we're okay. All right, so a budding yeast goes through this cycle. So we see right here we have a mature yeast, and it's going to start reproducing asexually with a bud on the top right there, and eventually it will let go of the daughter, and then it'll go back through. The daughter will go back through. This right here is the daughter cell right there, and it leaves a little scar there called the bud scar. And so oftentimes we'll see it, there it is, developing new bud. It sometimes can form the chain of buds, but those will eventually break off. Oftentimes under the scope we'll see something like that. So we call that a budding yeast. And we can see this is the daughter cell here on the left. There's the mature cell over there on the right. And if you take some yeast, if you've ever made bread, you know how you activate your yeast. So what we do is we go through a fermentation process where we take the yeast, we add a little sugar to it, a little bit of warm water, not too hot, and it will get it going and start bubbling and stuff like this. So if I had started that this morning, if I'd brought the yeast, then I would have started this morning. We could have looked at it under the scope, and probably by this time, it takes a little bit of time to have that reproduction take place, but we probably could have seen some of the budding yeast in there. But the bubbles that it's making is CO2, and CO2 is carbon dioxide bubbles, and that's the bubbles it puts into bread to make bread rise. That's how it works. Meanwhile, it's also producing alcohol, which is how fermentation is. Yeast makes wine and beer, I guess, yeah. So that's how the fermentation is a natural process there, also making the bubbles as well. All right, so fermentation, we can see here. Oh, wait, maybe I had a video to watch real quick. Yes? Yeah.
This is a video of the fermentation process on a microscopic scale. The ovals you see there are yeast in bread dough, and the big blue circle that's forming is a bubble of carbon dioxide being formed in the fermentation process. Here's how it works. The yeast that are in bread dough feed off the sugars that are also in bread dough. As they feed, they break down those sugars into alcohol and carbon dioxide. And once again, carbon dioxide are those bubbles you see forming there. Because carbon dioxide is a gas, it pushes its way out of the bread dough, causing the bread dough to rise. Now, when the bread dough is put in the oven, the yeast are killed and the alcohol evaporates away. And in fact, that nice smell that you associate with bread baking is actually due in part to the alcohol that's being evaporated during the baking process.
All right, so you can see here, the baking yeast usually is using something called Saccharomyces cerevisiae. And when it's mixed with water, it will cause fermentation, causing the bubbles to make the dough rise right over there. And that is, of course, a huge commercial importance. Okay, bread is one of the staples, right, that have kept people alive over the centuries. Other Ascomycota include the morels. Remember we saw that video last time about the false morel and that the real morel, when you cut it open, is completely hollow on the inside. They're considered a delicacy. I know in the Midwest you can walk around in your backyard and find them and pick them and eat them. Although, you know, I told you never to eat a wild mushroom because it could be one of those false ones, right? But apparently they're very good. I've never seen them for sale in our grocery store. I've never had one as far as I know. I don't know. So I've never tasted a morel, but they're pretty cool looking. We have the cup fungi as well. Look at these guys. So they form a little cup. Water will actually get in there, but the outside of them, let me turn the light off so you can see a little better. The outside of it, do you see it's kind of bumpy there? Mm-hmm. That's where the spores are on these. You know, so kind of think of the outside of the cup as the underside of a regular mushroom. That's where the gills are, but these have pores rather than gills. And then here we have these around here. That's called the orange peel, I think. Orange peel fungi, and it's in Ascomycota. This type is a parasite. I'm going to write this one down because at the very end of the lecture you're supposed to have written down some parasitic or diseases. So I'm just going to write oak wilt, so we'll know at the end to put that in. I did that instead of, I wanted to put it where they were, what group they belong to, and then when we get back to the end, add that in. Okay, but see, oak wilt, of course, causes the leaves to turn brown. They, of course, can no longer perform photosynthesis at this point, and therefore the tree will eventually die. Around here we have a problem with oak wilt in our live oak trees. In the Bastrop area we don't have as many live oaks, but out west we do in the hill country, and there was a time when a good number of those were dying from oak wilt. And so it's a fungal infection. It has to be controlled somehow, getting rid of the fungi. All right, next, Zygomycota. Here we go. Example would be bread mold. I actually have some bread mold here today we're going to take a look at. It's pretty gross. There's asexual reproduction when the stolon, remember the stolon is that part that reaches out to the side. When it lengthens and forms new filaments, that's a way of reproduction in Zygomycota. Another method is to make a sporangiophore from aerial hyphae. Remember all these words from last time? The aerial hyphae and all that business. I'll show you pictures here to hopefully jog your memory there. In sexual reproduction, there's a zygospore. So the zygote surrounded by a hard covering. And these spores are pretty much everywhere. The reason we call it bread mold is because we tend to leave our bread on the countertop. And guess what falls on them? These little spores. And the spores find a really, really good place to reproduce inside the bread, and therefore they grow in our bread. So pretty much, you know this probably, if you leave any bread long enough in the bread box or whatever, it's going to end up with mold on it, yeah? That mold came from the air. Say again?
What if you never took it out of the container?
Well, you have to take it out to get it, to eat it.
What if you didn't? Would mold fall from that?
Yeah, well, furthermore, if you had it in a sealed container that no air got into, but the problem is when you take it out, it gets exposed. So we're being exposed to it right now. Right now, there's Zygomycota all around us. You know anybody who's got an allergy to mold? My family, we have allergies to mold, sneezing and all that kind of stuff. And I'll show you that here at the end as well. So there's some bread mold right here. This one's called rhizopus. It's very common. I have a roll over here that has the mold growing on it. Usually this time of year, it's usually a different time of year. Since your book's been changed, usually it's at the beginning of the year. I'm always looking for bread mold. And this week I went over to my bread area and I found several examples of bread mold, sadly. I should have brought some cheese mold as well. So here we have what's going on here is, remember the rhizoid or the mycelium is growing inside the bread right there, okay? And then it's reaching up and the stolon can go across and then putting up a little aerial hypha, which has a sporangion on it, and the spores will go into the air until we infect something else. And that kind of musty stuff, the dusty stuff you see on there, that's the spores that have landed there. All right, but if you leave it undisturbed, you can actually see the hyphae sticking up. Now, this little bread I brought today was in a bag, and therefore we might not be able to see the hyphae standing up. But I'm going to show you a technique to actually get some of that onto a slide so we can take a look at it under our scopes today. We'll be making that slide. So you see here that even if you cut off the little part on the top of that, look at that's down in the bread. So if you got mold on your bread, I would recommend probably don't eat your bread, so throw it out. So we can see here that it's going through the process all inside your bread right there. Here's our rhizoid, our hypha coming up, a sporangiophore. We have our sexual part of it and our asexual part all making the rounds coming through here. We have our zygospore for the sexual part. Here's the sexual reproduction here, and making our zygote. but it also has the asexual rounds as well. Okay, so here's our strawberries. Yeah, so fruit is pretty susceptible to this kind of infection as well because it's sitting out. We leave the fruit sitting in the fruit bowl, and therefore the stuff's just going to land on it from the air. So you see what the mold's doing to it, what the fungus is doing to it, is digesting it. It's digesting it, and it's going to leave that liquid down there. I'm not sure how long that took for that to happen. Let's watch a little video on reproduction in bread molds. We'll see that different thing there.
Bread mold has several means of reproduction at its disposal. It can reproduce asexually by lengthening a stolon. Eventually the stolon will form a new mycelium, which will become a new mold. It can also asexually reproduce by releasing spores from a sporophore. Those spores will germinate into a new mycelium. Bread molds can also sexually reproduce when hypha from different mycelia fuse. Where they fuse, a zygospore will form, which will eventually germinate into a new mycelium. So bread molds can reproduce asexually through stolons. They can also reproduce asexually by releasing spores, and they can reproduce sexually, forming a zygospore. It's this mode of sexual reproduction that classifies them in phylum Zygomycota.
That's that word, zygospore, zygomycota. Myco, M-Y-C-O, is fungal. That means fungus. A mycologist is a person who studies fungi. Here we can see some fruit. I've never really figured out if that's a muffin, maybe. That's an orange for sure on the left, but I don't know if that's a muffin. Anyway, I wouldn't eat that for breakfast. This is in this group right here. It's called the pilobolus or the pilobolus, and it's kind of got an interesting little way of distributing its spores right here. You can see these little black dots on there as the spore sac. It's called the sporangium right there, and there's this like water-filled vesicle right here that'll get big and it'll burst. And when it bursts and explodes, it sends that thing flying. So here's a cartoon here. It gets big enough, it sends it out, and therefore the spore can be in a new location and grow a new organism over somewhere else. Okay, Chytridiomycota. That's your next group, Chytridiomycota. What you're looking at over here on the right is a potato that's suffering an infection from Chytridiomycota. Saprophytic in wet areas, feeding on dead plants. Some are parasitic, like this potato wart, it's called. And their spores have flagella, giving them motility to move around in a wet environment for survival. Of those things. Now, next week when we have a quiz, you might have some questions on some of these groups. Yikes, okay? So they're really hard words to remember there. Here's some more potato wart. You can see why I call it wart now. It looks like little warts. Here's microscopic views of these things. Here are their sporangiophore, or their little spores in there. Now, this one's pretty cool. Have you ever heard of cordyceps mushroom? Have you ever heard of this? Anybody you know into health food? Have you ever talked about eating mushrooms?
Mm-hmm.
Because cordyceps is one of the ones we're supposed to eat. I have it in my coffee. My coffee has cordyceps in it. And it's supposed to be really good for your brain and all this kind of stuff, but it turns out it's pathogenic.
Does it have a different name?
No, that's it, cordyceps.
Never heard of it.
But it's only pathogenic to bugs. So the bugs are really afraid of it. Okay? But I have to show you. This is really creepy. That's like a zombie situation going on there. It grows inside the brain and makes the insect do its will by crawling to a high spot and holding on so it can send out its spores. And apparently those cordyceps mushrooms are good for us to eat as a health food, so that's crazy. Is this the same? Is this the same one? I think it must be the same one.
The success.
Okay, I don't know why sometimes they go in there twice. I don't know how that's. Okay, now we're on Deuteromycota. The example would be penicillin. We're going to be looking at Alexander Fleming's work that he did in the 20s, and how he was able to isolate something that would become one of the world's greatest medicines called penicillin antibiotic. Examples would be in cheese. Roquefort, I was supposed to pick up some blue cheese yesterday. Forgot that too. So I'm really having some brain problems here. Maybe I have a zombie fungus in my head. I don't know. But I remember forgetting all sorts of things. But it's okay. We have plenty to do today. They're called the imperfect fungi because we don't know how they reproduce. So when it comes to reproduction on these guys, even now, we don't know how they do it.
So we make it into a medicine and we don't even know how it fully works.
Right, we just know that it does work.
Okay.
Right, so, but we don't know the mechanism by which it works, yeah. The mechanism by which these guys reproduce. We know the mechanism by which the drug works because it actually kills bacteria by punching a hole in their cell membrane. All right, I believe that Deuteromycota means imperfect somehow, that word. That's where the imperfect comes from. All right, so Penicillium notatum is one of those, and that's what it looks like under the microscope. We will be looking at this under the microscope on prepared slides as well, and it should look something just like that, and you'll be drawing that. All right, so this is showing a agar plate that Alexander Fleming used, and he went on vacation and came back. Remember I told the story, and how he was very upset because there was a mold contaminating his dish, his petri dish, and he was going to have to throw everything out. But then he noticed something interesting, this little halo between the mold growing on the bottom here and the growth of the bacteria. That's that smear looking thing right there. And he said something in the mold is killing the bacteria. And they went with that and found out the secretion. They turned that secretion into medication, and now we have antibiotics. And because of that, we found out other things that produce stuff that kills bacteria. And it's very fortunate for us because sometimes we really pretty much, I'm not going to say we've tamed completely bacterial infections, but we do have an upper leg over bacterial infections. So much so that we've overused the antibiotics a little bit, and some of our bacteria are adapting and mutating to become stronger, so that we'll need newer and better antibiotics as we go. So the bottom line is we shouldn't use antibiotics unless we really, really need them. And if we need them, fortunately, we have them. Here is Penicillium roquefortii, which you can see when you talk about blue cheese. Y'all like cheese? You know, cheese is formed through fungal action. And this one is a particularly good, it shows it really well. So what I would normally do is buy some blue cheese and leave it out for a couple of days and let it ripen a little bit, and we would make a slide, and it gets really stinky. Some people really, really like stinky cheese. My preference is for cheese that's not stinky.
I don't like cheese.
Do y'all like blue cheese? I do not care for blue cheese, but I have friends who love it, blue cheese dressing, all that kind of stuff. It's got sort of a rotting kind of a flavor, if you ask me. This is what it looks like under the scope. Pretty cool looking. All right, Myxomycota. Is this a real one, or is this that last one? Oh yeah, this is no longer in the group of fungi anymore. Apparently, according to our book, they have fully moved it over to the protist kingdom. But lots of people still see it behaves like a fungus, but it feeds like a protozoan. That's why I decided to put it over there, I guess. What they need is a moist environment. In the feeding stage, it exists in a blob called the plasmodium. The fruiting bodies are similar to sporophores that release the spores. Okay, I'm going to go ahead and watch whatever we have here, even though it should go in the previous module, or I guess previous week.
On these rotting tree trunks, something is alive. They can stand upright and produce spores, but they're not fungi or plants. When they're hungry, they spread across the forest, chasing food, but they're not animals. They're slime molds. A slime mold exists at the boundary between liquid and solid. Each one is like one big cell, and it moves in a very strange way. See how it pulses a little bit forward, a little bit back, spreading and searching for bacteria or fungi to eat. But hold on, today we have someone else who moves in strange ways. The host of It's Okay to Be Smart, Joe Hansen. Oh, hello, Joe.
Hey there. So from down here, it's pretty obvious that slime molds don't have legs or any appendages. They move just by changing their shape.
Scientists want to know how this works. So they brought a type of slime mold into the lab and fed it some oats. At the University of California in San Diego, they put a piece of slime mold under a microscope, watched its weird pulsating movement up close.
That motion is triggered by chemicals moving around inside it.
Water, moving in waves, carries tiny calcium molecules from back to front and back again. The molecules are too small to see. They're not those circles. Those are the slime mold's many nuclei, among other stuff. But scientists can track the calcium flow. In this video, it appears in light blue. The calcium triggers a reaction that makes the cell wall contract.
The slime mold squeezes itself, almost like a water balloon.
That contracting and sloshing helps the slime mold inch forward, bit by bit. And it kind of begs the question, could we build something that moves that way? I mean, usually we think of robots as solid, hard-edged machines that roll or walk from one place to another. But what if they were fluid and powered by chemical reactions? Like this Japanese walking gel. Maybe this is a really early step towards some hypothetical robotic goo that could squeeze into the narrowest parts of your body. Hey Joe, imagine one of these inching through your arteries, helping clean them out as it goes.
That might be kind of, creepy.
Yeah. I don't think I'd want them in my arteries. So there's one, look how pretty. And the next day it'll look very different because it will have moved, right? And there's another one. Here's this close up. All right, so these, look how they look a lot like fungi, don't they? Those look like club fungi right there. And then of course from last time we talked about this one, remember that one's called dog vomit. And here are some of the fruiting bodies coming off one. Those look like mushrooms. So I still see how they could fit into the kingdom fungi. But according to our book, we're going to go with what our book says. It says it belongs in kingdom Protista. All right, here's some along the edges of a leaf right here. All right, life without fungi. You have on your next sheet something about what would life be without fungi. What would it be like? We would have no G's. So write no G's.
That would be good.
That wouldn't be so bad. We would have no mushrooms to eat. Not too bad. In your next blank, I didn't put a sign up here, but put up no bread. No bread. Or things that need yeast to rise. No wine, no beer. You know? We wouldn't have penicillin. And a lot of medications come from fungi. Others come from plants and things like that. We would have a lot of piled up leaves and dead things because it wouldn't be decomposed by fungi. So piling up dead bodies, you can write including leaves. Lovely. And then you have one more blank?
No.
No?
Well, I put no bread.
What
in
about-
one of them.
Okay, that's good. Because the other one was plants wouldn't be as healthy, but I don't know about that
What
one.
about these?
We're going to be filling in blanks here and there just a second. Oh, here we go. Fungi are decomposers that allow ecosystems to stay balanced. That's something you're filling in right there. Decomposers in one of your blanks. Seems like an awful late to be talking about that in our lecture. I think I've mentioned it many times, right? Okay. And then lichens. A lichen is a symbiotic relationship between a fungus and an alga. So it's a fungi and an algae working together. They benefit each other, so it is a mutualistic relationship.
One needs the other.
They benefit each other. They live together beautifully. So they reproduce a sporidium, which is a unique spore case containing spores from both organisms. Very unusual that would happen. You can see here we have our fungal hyphae. We have algae living in the middle right there, and more fungal hyphae. The fungus is supporting and protecting the algae cells. And what do you think the algae cells are doing for the fungus? Think about what algae does. Namely, it's green. It's making food. The fungus, it's like they're farming it. It's like the fungus is farming the algae. And they're both benefited because the algae can live well and be protected from any, like just being squished or washed away or something like that. Let's watch a little video on it.
A horticulture extension agent with the Clemson University Home and Garden Information Center.
That's 4-H.
Today we're going to take a look at lichens. At the Home and Garden Information Center, we get lots of calls from people who believe that lichens are killing their trees and shrubs. The good news is that these lichens are not harming their plants in any way. Instead, they are taking advantage of a spot that just happens to receive adequate sunlight. Now, it is true that lichens are often found growing on ailing plants, which makes it hard to believe that they're not to blame for their decline. But if you do take a look at trees growing throughout the forest, you will see that they're often found just growing on healthy trees in those areas. Now, if we take a look at this paper bush shrub, you will see that it has very thin foliage, and that creates an environment where sunlight is going to move into these inner branches. It's an ideal environment for lichens to move in. However, it is a sign that this plant is performing poorly, and they need to look at the culture and see what needs to be changed to thicken up the foliage on this plant to get the lichens to go away. Lichens are not parasites, and they don't cause any harm to trees or shrubs in any way. In fact, when you're out on a nature walk, you can find them growing in some unusual places, like on stone structures of arbors, wooden areas on arbors, park benches, or even on rocks. Lichens are plant-like organisms. They aren't a moss and they aren't a plant. In fact, they're actually an alga and a fungus growing together in the same body in a symbiotic relationship. The fungus provides the structure to the lichen, and the alga provides the food through photosynthesis. There are many different species of lichens, and those lichens are divided up into 4 primary groups according to their body type. First, we have the foliose group, and the foliose group is a 2-dimensional type lichen. It has a very lobed, leafy appearance to it. And then there is the fruticose group, and the fruticose group is a very 3-dimensional, very shrubby and highly branched type lichen. It looks like a little shrub. And there's the crustose group. That one is very tightly held to whatever surface it's growing on, whether it's a rock or a tree, and it's kind of scaly in appearance. And then there's the squamulose group, and that one's kind of a mixture of the foliose and crustose group together. Lichens need 3 basic things to become established. 1, they need an undisturbed area to become established. 2, they need enough time. And 3, they need clean air. If you find that you have a large population of lichens on trees and shrubs in your landscape and you would like to reduce those, certainly by improving the health of those trees and shrubs, you're going to help to reduce the lichen population and they will start to go away. In general, trees and shrubs need to make sure they have good drainage, the right amount of sunlight, and proper nutrition. For information on your specific plant species, you can find growing details plant by plant on our website at www.clemson.edu/HGIC.
Okay, so while we have that mutualistic relationship between the fungus and the alga, we also have it with plants. So this is called mycorrhizae. And so when we have a fungus, mycelium, and a plant's root system growing together in a beneficial way, we saw last time the Haustorium, which was a parasitic invasion of a fungus into a plant. Here is a symbiotic mutualistic relationship. So the mycelium will take nutrients from the root while it collects minerals from the soil, and the root can absorb the minerals. So they're working to help one another. So we can see here we have the plant part and then the mycorrhizal part there. So these fungi are beneficial. They form this symbiotic relationship with the roots. Keep in mind that that video we saw about the lichens are also not a problem. They are not parasitic at all. It's just what she was saying about some trees, if they have really bare spots on them, maybe the tree is unhealthy, but it's not the lichen causing it. That's all I want you to say. Same here. This is not anything causing a problem for the plant. Now, I haven't watched this video in a while. Let's see if it's still on.
Mycorrhizal fungi play key roles in the nutrition of plants. This is critical when it comes to tightly bound soil nutrients such as phosphorus. As non-mycorrhizal roots grow into the soil, most of the phosphorus is unavailable. Compare this to mycorrhizal colonized roots. Here, tiny mycorrhizal filaments called hyphae explore significantly greater volumes of soil resource and excrete powerful enzymes that unlock phosphorus from soil surfaces and act as a conduit directly back to the plant. The result is improved phosphorus uptake and nutrition.
Interesting music there. Yeah. So All right, so these are some leafcutter ants. I'm bringing them in here because that video we watched about the cordyceps earlier was kind of, the beginning of it was talking about these guys. What they'll do is they'll take these leaves. People said, well, they thought they were taking the leaves to go eat them. Well, they weren't. They were taking the leaves actually to farm them. And what they were farming on the leaves was fungi. So they'd grow a fungus garden right here, and they use the fungi to feed their brood in the ant colony right there. Look at some of these ants, the soldiers. Look at that. Man, they're huge and very scary looking. And then our workers are over here cultivating the fungus. All right, and now this brings us to bad stuff, right? Do we have, is there another blank on your book about bad stuff of fungi?
Well, there's 2 more blanks on it.
What's one of the blanks? What's the top blank?
It's really this. It's like the word, like the lines at the bottom.
What does it say?
It says relationship between an algae and then blank, and then
Okay,
some
that's,
fungus is pathogenic such as.
well, you missed that one. That was,
Oh, well, I didn't see.
yeah, that was the lichen. Right here. Lichen's a symbiotic, right? This one?
Yeah, and then there's an extra at the end though. It's not up there.
Say what it says again.
Relationship between an algae and a fungi, and then it's period, and then some fungus is pathogenic such as.
Oh, okay, that's what we're talking about. The pathogenic ones, such as oak wilt. We wrote down that earlier. And if you want to say just mold growth and the black mold, that could go on that line. This is a house, this is somebody's house, and I'm thinking there must have been like a plumbing leak upstairs or something, because apparently the drywall got wet. And then this mold is growing on there, growing through the drywall down in there, and you see this. If you have a problem like this, the people need to move out so we could have what's known as mold remediation. And that means they have to take all that drywall out of there.
House at that point.
They probably just have to gut the whole house at this point, because the mold will keep growing. So we found, my mother lived in a little house across the yard from me, a little tiny house, and after she passed away, my son was going to move in there, and they noticed some mold growth in the house, and we found out it was in the air conditioning vent, and that's very common. And so the idea of going in and cleaning out the air conditioning vent, all the mold people said, oh, that's just going to disperse the mold further. So what we did is we sealed those off and put in mini splits. You know what a mini split is? Like the little air conditioner units that don't have the whole central air. So we put those, you see those sometimes in little Airbnb, little hotel rooms. This is a tiny house, so we put it in there like that. But we were concerned about the whole house getting eaten up with mold, and we were able to staunch it off. And now keeping a place dry and cool, that'll keep the mold down. So that's why we use our air conditioners, right? Also to be comfortable. All right, what about allergies? That can be another thing that they're bad for. People who have allergies to fungi, mostly mold, and it gives you the itchy eyes, the runny nose, the headaches, things like this. And that's when we can have allergies to many things, but mold is one of those that gives us that. There are also fungal diseases that can be detrimental to humans as well. We talked about that a little bit last time, about the ones you get from the cat, cleaning the cat box and stuff like that. Oh no, sorry, I should have warned you about this. Toenail fungus. So think about, we keep our feet in our shoes, it's dark, it's kind of moist. The fungi love it. I will tell you, if you have fungus on your toes, it's not coming from outside, it's coming from inside of you. So trying to heal it by putting something topical on it's not that effective. Okay, time for the lab. We're going to do some, so I didn't fill out the rest of my deal, but you should have had something in each one of those boxes, right? Here's what I put down in my ask on my code. I never filled out the rest of it because I just was too lazy to do that. All right, so grab your scope. I left one near you. You should have some little circles to draw some microscope work, yes? Somewhere, either last time or this time?
Didn't you say the F meant something?
No, D for Doug.
Oh, yes, okay.
All right, so what I have here is I have penicillium and aspergillus. You can draw either one, so I'll give you one of these. You draw this one, you draw this one. And then both of you have to, though, draw this one. You have to share this slide. This is the budding yeast. So start with this one. So then when you're finished with this one, you can send it over to Preston, and then you can draw this one. Let me see. I thought we had some circles and never. No. Maybe there was some. Yeah, I guess you're just going to use this lab report on page 82. Draw some circles on there. And then we're going to make our own slide out of our bread mold, which is pretty darn gross. Oh, and I do have some lichens for you to take a look at. These are just from the little branches off my driveway. We have lichens everywhere.
They're everywhere at the house. I didn't know
Yeah.
that's what they were.
Yeah, so you can see here, and this one's kinda cool looking, has the foliaceous ones and the filamentous ones, both on there.
Yeah, they're everywhere in our trees.
So find the best looking view that you can, whichever level is the best. On your experiment, right, looking at fungi, and you're gonna also draw some of this thing, a lichen, and then you're also gonna do the bread mold one. I'll pull it out. It's pretty nasty.
This is a hard thing to draw.
Are they pretty cool looking? The budding yeast may not be that cool looking, it should just look like the little figure eight things, right? That's what it looks like?
Yeah, it's a crazy shape. You know, it has like a tail. You wanna switch? Sure.
You have another one here, yeah.
Switch them.
Okay, yeah, you can draw the one he's drawing, that's fine.
Whoa, this is way cooler than mine.
Yes, cause you were just looking at budding yeast. And that's fungi, and they're really cool looking, so.
Cute.
Remember to tell me the magnification at which you're looking at that,
Oh, yeah.
and label what it is.
It's just a 10 different times.
Is that what that is?
Huh?
Is that what that, 10 times?
I think it's
100, yeah, 10
yes,
times.
it's 10. Is that 1,000? No.
No,
So this is 10, right?
that's, yeah, so 100, no, that's 4, so
Oh,
40, and then
Oh.
10 is 100, and this is 40, so it's 400.
okay.
If it's the highest one, right? Is it the highest
Yeah,
one?
it is.
Yeah.
You have any more?
No, that's it, unless you wanna check that one out, it's the same thing, it's similar to the one you're already looking at. Alright. Now I'm gonna teach y'all a technique of making a slide, and I promise you this is what they really do. It's not like, just like a hack or anything, it's a genuine, technique. So we're still Nutri-Scopes there. So here is our piece of bread, believe it or not. Yeah, I was eating that at one point. And it got kind of squished back in the back of the bread box and I forgot about it or whatever. So you can see I've got several different molds growing on there. So I've got this greenish one here, it's an orangey one here, a yellow one here, a different green right there. And if you look right here, this white one, do you see how that you can see the aerial hyphae coming up right there. Yeah, you can see they're sticking off. Now up here they're not sticking out so much because they were in a bag, and the bag was kind of keeping it down. All right, so what I'd like y'all to do, you're going to make a slide. I gave you a blank slide, and what you're going to do is I'm going to bring it. You're going to give you this little piece of tape right here, and you're literally going to take this tape down
Press it on.
and press it onto one part of the roll, and then tape it right to your slide. Okay, so I'd like one of you to take maybe the green, and then somebody to get this white one right here. So here, take it with both your fingers on the edges. I've got my fingerprints too. I just washed my hands. And yeah, just pick a side and then leave. And Preston, here's your piece. So as long as you use kind of a pretty clear tape, yeah, you don't need much. You can just barely touch it. And look how much you got on there. So Preston, I'm gonna try to get you to get that white stuff right there.
Right there.
Yeah, and see if it comes off now. Oh my goodness, yeah. So stick it all right down there, and then you're gonna take a look at that in the scope and see what you see. You're gonna have to go to the edges of it because you're gonna have a ton of stuff in there.
Oh my god.
You see a bunch of stuff, like a giant forest of stuff in there? And notice, you know, we didn't even stain this, right? We didn't need to because it had a really pretty much its own color to it. But try to go to an edge or something,
I don't think I can draw
see if you can see see some detail versus, oops, I'm still recording. No, I finished, okay. No, I'm still recording.